All right, welcome back. Today we're going to make a transition away from just talking about cell communication and instead talk about the cell cycle, cell growth, cell division. What does a cell go through during its lifetime? And the lifetime of a cell is very different depending on the type of cell that's in question. Some have a pretty straightforward life cycle, and others may go in and out of different stages of their life cycle as they age. So, we're going to talk about general and more specific cases of what happens in the life cycle of a cell. So, first and foremost, why might cells need to divide? Or, in other words, why might cells need to replicate, make new cells, right? Cells will make new cells by dividing. Now, there's three more main ways why cells might need to divide or why cells might need to make new cells. And that's one for reproduction. So, like asexual single-celled organisms like this amoeba here, that's how they reproduce. They divide and they've made a carbon copy, a clone, a new cell. So, where there used to be one, now there's two. For asexual organisms, or asexual single-celled organisms like this amoeba, that is what they do to replicate. Additionally, cells might need to divide for growth. So, for example, as an organism grows from small to larger throughout its lifetime, it needs to continually make many, many, many new cells to have tissue to grow larger. Even at the very beginning of life, when we start with a fertilized egg, all of the um, all of those cells will eventually divide hundreds and thousands of times to form the 10 to 100 trillion cells that are in a full-grown living organism like a human. And lastly, cells might need to divide to repair and for new old, dead, or injured cells. Just like this lizard has lost its tail, maybe in a violent encounter, and it will regrow back. It regrowing its tail will require many new cells to repair the old ones that had been removed. So cells divide, cells make new cells for these three specific reasons. So if a cell is going to divide to make a new cell, the new cell is going to need some important information or some important resources to be able to live and maintain homeostasis. One key thing is that it will need an exact copy of the same genetic material that the original cell, we'll call it a parent cell, started off with. So if this cell in the microscopic image here was going to divide and going to make a new cell, it would need to ensure that every single chromosome, every single piece of DNA that it contained was copied exactly into the new cell. Secondly, or that process of making sure that all of the same genetic material in the parent cell uh, gets passed on to the new cell, we'll call the daughter cell, that is mitosis. That process is taking care of mitosis when chromosomes are duplicated and then divide equally amongst our two cells. The new cell will also need organelles and cytoplasm and membranes and enzymes to do all the various functions it will need to do. And after mitosis occurs cytokinesis is the process of these organelles and cytoplasms and membranes and enzymes being divided amongst the two new cells. But mitosis is, or the actual division and separation of chromosomes is only a small portion of a cell's life cycle. Most of the time, 90% of the time, a cell is in a stage of its life cycle called interphase. And this is where the cell grows, the cell goes about its specified function, and it produces new organelles and enzymes. And it also replicate its chromosomes or make a copy of its DNA in preparation for dividing. But most of the time, 90% of the time of an average cell, life is spent in interphase. Now, if you look at this picture up here, the gray parts together are called interphase, and we subdivide interphase into three smaller chunks. This G1 phase, this S phase, and this G2 phase. The purple parts are mitosis for the lighter purple part, and then cytokinesis, the darker purple parts. Remember, the mitosis, the lighter purple parts, that is when the cell separates and the DNA, the chromosomes are divided equally to make sure each cell has an exact copy. The dark purple part, this is cytokinesis. This is when the organelles and the cytoplasm are divided amongst the two cells. So, like I said, 90% of a cell's like cycle is in interphase. And this is where it's doing its everyday job. It's preparing in case it will be asked to divide to duplicate. And in fact, that's what occurs in this S phase. This is where a phase of interphase where DNA is copied so that the cell has two copies of DNA in case it might need to imminently divide. But generally, in interphase, this is when the normal life cycle of the cell is occurring. Now, um, m- all cells have a life cycle, but it might be different depending on the type of the cell, right? So let's start maybe from what we call the birth of a cell. So a new cell is formed here in this blue part from the mitosis, the division of an old cell. Now, depending on the type of a cell, it might take a couple of different paths. One, a cell maybe like a skin cell or like the cells in your stomach lining that grow and are used up and divide really fast. It might go down this green path. The cell might grow and mature and then divide again. So that means the might go through G1 phase where it is gathering resources and preparing to duplicate its DNA. It might then go through the S phase where it duplicates its DNA to divide. It might go through G2 phase where it's gathering resources and preparing for division, and then it might divide again right away. So things like stem cells and blood cells and epithelial cells um, do this kind of pattern. They are continually are maturing and dividing and maturing and dividing, and they replace one another quite quickly. For example, it takes, maybe 7 to 21 days to replace every single cell in your stomach lining because they replace each other very, very, very quickly, right? Other cells might go through this stage, same type of stage, but do it the whole process might be much slower. So instead of all of the cells like in your stomach lining being replaced every 7 to 21 days, your skeleton the bone cells might go through the same process, but much slower. So that would take, 15 to 21 years for every one of those bone cells to be replaced, to replace an entire skeleton. But it will happen. So some types of cells will do this faster, some will do it slower. But one option is for this kind of slow or quick, but continual division. Another option, though, for example, like nerve cells in your brain and muscle cells, once they are formed from the division of and specialization of stem cells, they will grow and mature, but they will never divide again. They will never divide again. Once they are formed from a stem cell dividing and then specializing, differentiating, they will never divide. So your neurons do not make new neurons. Or muscle cells do not make new muscle cells. when this happens, we say that these cells, after they're formed and enter G1 phase, they transition to this what's called G0 phase. It's kind of like completely outside of the cell cycle, this kind of limbo where they're staying dormant, never to re-enter the cell cycle again. There are some cells that are only in very specialized tissues, like the liver, that will grow and mature and go into this G0 phase where they've exited the cell cycle until something happens. So, like a liver cell will grow and divide, will grow and mature and enter G0 phase until the liver is damaged. And then these liver cells can somehow, inexplicably, we're not positive how this happens, but they're capable of jumping out of this G0 phase and entering the cell cycle to make new cells to repair the damage, and then jumping back out into G0 phase again. You can replace or regrow a whole liver from about a quarter of it. This is a huge area of research because we're really not sure how this happens or what the stimulus is to cause this jump from G0 to G1 phase, but it can do this. Scientists are super interested in how this works because it would be awesome to know to be able to potentially stimulate like nerve cells or muscle cells to repair injured or dead nerve or muscle cells by stimulating them to jump back into the cell cycle like liver cells. But to this point, we're not positive of how to do that. So here's another picture just of interphase and the three parts that make up interphase G1, S, and G2. It summarizes slightly about what is happening in each of the phases. And then we have this, the M phase, the mitotic phase is right here, kind of in the light yellow color. So now we're going to talk about a little bit of the characteristics of the cell, what's going on in the cell in each of the phases of interphase and then mitosis, and how to be able to determine what phase a given cell is in. So in interphase, the biggest idea is the nucleus is very well defined, and the DNA kind of looks like a stringy mess called chromatin in the nucleus. So notice how this nucleus is very well defined. There's a sharp nuclear envelope. And then inside the cell, you can't see individual X chromosomes. These are chromatin at this point. This is in interphase. This is in interphase. In interphase and the S phase, this is when copying or replicating of DNA occurs because each cell needs to replicate its DNA in order to have a complete and exact and correct copy for the two daughter cells. For example, so in human cells, each time they divide, about 3 meters of DNA needs to be duplicated, right? And this happens so efficiently that there's only about an error rate of one error per every 100 millions of base pairs, right? To put that in perspective, there's about 3 billion base pairs in a mammalian genome. So for about every cell cycle or every replication, there's about 30 errors that happens per cell. And these errors are mutations. Mainly they happen in somatic or body cells. Then when we get to mitosis, so we've gone through interphase, we're prepared, ready for it to divide, we separate mitosis into four smaller phases. Now we need to know roughly, not every single excruciating detail, but roughly the important things that happen in each phase. Here is an overview of each of the phases. Note, some texts make a distinction of pro-metaphase from prophase and metaphase. For the purpose of this class in the exam, you do not need to know the separate events of pro-metaphase, just prophase and metaphase. There's also a really nice picture with descriptions in your book in chapter 9.2, I believe, that is very much like this, but also includes some descriptions. So after interphase, there comes prophase. And this is when the big idea the biggest ideas I have here in green. This is when the chromosomes become visible. The chromatin, that stringy mass of DNA that was in the nucleus, condenses, becomes more dense, and now you can see the nice X-shaped chromosomes. Now we can call them chromosomes. The centrioles or centrosomes move to opposite sides of the cell, and these projections made out of microtubules called spindle fibers start to extend their way out. And the nucleus again, the nucleolus or that dark area where ribosomes are being made, disappears, and the nuclear envelope, that plasma membrane that surrounds the nucleus, it breaks down or breaks apart. Then we transition to metaphase, right? We transition to metaphase. And the idea in metaphase to transition to metaphase is in metaphase, we're going to want all of these chromosomes to be lined up across the middle of the cell so that they can be split, so that each daughter cell can get one of each. So in the transition to metaphase, our spindle fibers are attaching to the middle of each chromosome. And now the middle of each chromosome is called the centromere. And at the centromere, there is a special protein called the kinetochore where the spindle fibers attach. So this picture, you see the spindle fibers attaching the kinetochore from both sides at the centromere. And so we see that happening and the chromosomes beginning to line up across the middle of the cell. When they have finished lining up across the middle of the cell, we are now in metaphase. It's one of the easiest phases to spot on a microscope picture because all the chromosomes are lined up. Each chromosome is lined up properly so that when these chromosomes are split, are separated, one half or one copy of each chromosome will be inherited by the new daughter cell. The next phase, anaphase, occurs when the chromosomes that start to separate, when each sister chromatid, that's what these halves of each chromosome are called, start to walk along the spindle fibers in opposite direction and pull apart at the centromere. So anaphase away, the chromosomes move away from each other, pulled by motor proteins that walk around along these microtubules of the spindle fibers, and they're moving farther and farther and farther apart towards the opposite poles of the cell. So here's a closer picture of the separation of chromosomes in anaphase. So here we have one chromosome with two halves, two identical sister chromatids, like in metaphase. And then in anaphase, when they start to walk in opposite directions along the microtubules of the spindle fibers, they have separated into two daughter chromosomes that will start to will be the new genetic information of each cell. Here's a close-up picture of the centromere and kinetochore of the uh, the chromosomes, and you can see how the kinetochore has a little motor protein in it that actually walks the chromosomes back along microtubules towards opposite sides of the cell. So the tubules don't pull the chromosomes apart. That is a common misconception. What happens is the motor proteins in the kinetochore walk the sister chromatids back in opposite directions along the microtubule spindle fibers and thus separate and rip them apart. So they're a shortening and dismantling of the microtubules by the kinetochore motor proteins. Lastly, in telophase, each chromosome has now arrived at the opposite side of the cell, the two poles, and two new nuclei, one around each complete set of DNA, starts to form. So the nuclear envelope starts to form again. The chromosomes start to disperse and become less dense back into chromatin, and the spindle fibers disappear. At this point, cytokinesis can begin to occur, or the organelles in the cytoplasm of the one original cell can get evenly and equitably divided so that both daughter cells now have everything that they need to function as new cells. In this slide, we're pointing out the pinch off part called the cleavage furrow starting to form, which will eventually be the separation point between our two new chromosomes. And this occurs um, because of actin and myosin fibers right here at the interface between the two new cells that will start to tighten and tighten like a drawstring, closing up a bag, and eventually pinch off the two new daughter cells from one another. Here's a picture of the cleavage furrow here forming in an animal cell that is dividing into two. Here are some other pictures of interphase, prophase, metaphase, anaphase, early telophase, and late telophase in some whitefish blastula, so some very early cells in whitefish. And you can see dyed in dark purple contrast the DNA, the chromosomes of each cell. Right? So that's what we want to make sure that we can talk about today. And we'll finish up with talking about how the cell cycle is all regulated next lesson.